hormones are present in blood in very small concentrations, yet they produce profound biological changes. The action of a hormone occurs through a cascade of events in which the signal is amplified at a number of stages. So, glucagon receptor complex is there. Inactive adenylate cyclase turns active adenylate cyclase. Then ATP turned into CAMP. There will be some kind of amplification. Then inactive protein kinase A, active protein kinase A, there again going to be some sort of amplification. Then inactive phosphorylase kinase B, active phosphorylase kinase B, amplification, inactive phosphorylase, active phosphorylase, amplification, glucagon, glucose 1, glucose. A molecule secreted by some cells that controls and regulates the activity of certain cells or organs. The hormone acts on its target cells which possess specific receptors for the hormone. The target cells may be distant or nearby. Depending on the distance between the target cell and the hormone secreting cell, the hormone may be endocrine hormone, paracrine hormone, or autocrine hormone. The endocrine hormones are secreted by endocrine ductless glands and act on distant cells. The paracrine hormones act on cells in near vicinity of the hormone secreting cells. Then the autocrine hormones act on the cell secreting the hormone. Hormone receptors. Target cell possess receptors for hormones. Chemically, the receptors are proteins. A receptor is specific for a particular hormone. It has at least two domains. So, target cell possess receptors for hormones. Chemically, the receptors are proteins. A receptor is specific for a particular hormone. It has at least two domains, a recognition domain and a signal domain. Chemically, the receptors are proteins. At least two domains will be there, a recognition domain and a signal domain. The receptor may be located in the cell membrane or inside the cell. The intracellular receptor may be located in the cytosol or in the nucleus. A given cell may possess receptors for several hormones. So here we could see a picture which depicts receptors. 1, 2, 3, 4, 5, 6 are hormone receptors. Nucleus, inside nucleus, in cytoplasm, everywhere. The number of receptors is not, so two regions are there. Which are they? Signal domain and recognition domain. Recognition domain and signal domain. The number of receptors is not fixed. The receptors can undergo upregulation, increase in the number of receptors, and downregulation, decrease in the number of receptors. Then classification of hormones. Hormones may be classified on the basis of chemical nature, solubility, location of receptors, and nature of second messengers. First, on chemical basis, hormones can be divided into proteins, peptides, and amino acid derivatives. Then steroids. And earlier we mentioned another category which is amino acid related hormones derived from tyrosine. And the basis of solubility, hormones can be divided into hydrophilic hormones, proteins, peptides, and catecholamines, hydrophobic hormones, steroid hormones, and thyroid hormones. The classification based on the location of receptors and nature of second messengers is more comprehensive. On this basis, the hormones may be divided into two groups, group 1 hormones and group 2 hormones. Group 1 hormones. These hormones are lipophilic. Lipophilic in the sense lipid loving. Group 1 hormones are lipophilic. Examples are steroid hormones, thyroid hormones, and calcitrol. Being lipophilic or lipid loving, they require carrier proteins to transport them in blood. The receptors for glycocorticoids and mineralocorticoids are present in the cytosol. The receptors for estrogen, progesterone, thyroid hormones, and calcitrol are present in the nucleus. Mechanism of action of group 1 hormones. We mentioned the receptors of group 1 hormones are intracellular. Being lipid soluble, the hormones can easily traverse the cell membrane. The hormone bind to their intracellular receptors. The hormone receptor complex is mobile. The hormones bind to their intracellular receptors. The hormone receptor complex is mobile. It does not require any second messenger to carry the signal. Then the receptor possesses one domain to recognize and bind the hormone, another domain to recognize a specific sequence in the DNA. That DNA sequence is called hormone response element. Hormone response element. So there are different response elements for different hormones like glucocorticoid response element, mineralocorticoid response element, thyroid hormone response element. So the Hormone response elements are located just upstream of some structural genes and their promoters. The hormone receptor complex goes to the nucleus and binds to the HRE. HRE in the sense hormone response element. This increases the transcription and translation of specific genes. Transcription and translation of specific genes. Okay. Then the protein synthesized produce the biological effects attributed to the hormone. For example, glucocorticoids increase the expression of genes encoding gluconeogenic enzymes. Increased synthesis of these enzymes increase gluconeogenesis. Here we could see a picture which depicts all this. Hormone is there, receptor is there, hormone receptor complex is formed, which produce some sort of response by reacting with the HRE, hormone response element in the DNA inside nucleus, later producing an mRNA. Then ribosome is there, which produce protein which lead to a biological effect. Okay. Then group 2 hormones are there. Group 2 hormones include proteins, peptides and catecholamines being hydrophilic. Just remember, they are not lipophilic, they are hydrophilic. That means lipophobic. They do not require carriers to transport them in blood. Their receptors are transmembrane proteins. The hormones bind to their receptor on the cell surface and does not enter the cell. So this hormone binds to its receptor on the cell surface and does not enter the cell. A second messenger is required to carry the message inside the cell. A second messenger is produced by an effector. Mechanism of action of group 2 hormones. From now on, things are going to be a bit, more complex. So I need you to get into this topic. Just carefully listen. Okay. So on hormone receptor binding, a signal transducer is activated. The signal transducer goes to and activates an effector. The effector produces a second messenger. The second messengers include cyclic AMP, cyclic GMP, CA2 plus, and phosphoinositides, tyrosine kinase. CAMP. We are going to learn about CAMP in detail. The hormones using CAMP as a second messenger include TSH, ACTH, FSH, glucagon, epinephrine acting through alpha 2, beta 1 and beta 2 adrenergic receptors. So the hormones using CAMP as a second messenger include TSH, ACTH, FSH, glucagon, epinephrine acting through alpha 2, beta 1 and beta 2 adrenergic receptors. CAMP is produced in the cell from ATP by adenylate cyclase, a membrane-bound enzyme that is normally inactive. ATP, adenylate cyclase, CAMP plus PPA. ATP, adenylate cyclase, CAMP plus PPA. Binding of the hormone to its receptor activates adenylate cyclase. When concentration of CAMP in the cell increases, this produces the biological effects. Most of these effects occurs due to CAMP induced activation of protein kinase A. So as per this slide, we could see ATP turned to CAMP plus PPA with the help of adenylate cyclase. So binding of the hormone to its receptor activate adenylate cyclase. Then concentration of CAMP in the cell increase. This produces the biological effects. Most of these effects occur due to CAMP induced activation of protein kinase A. For example, activation of protein kinase A in liver by glucagon increases glucogenolysis, decreases glycogenesis. Then adenylate cyclase, which is an effector, is not in contact with the hormone receptor. The signal is carried from the receptor to the effector by the signal transducer, that is receptor effector coupling. The signal transducer is a G protein. So here we could see this G protein, alpha beta gamma subunit, DP bound stage. Inactive stage is GDP bound stage. Okay, receptor is there. So G protein is a trimer. G protein is a trimer made up of alpha subunit, beta subunit and gamma subunit. The alpha subunit has a site which can occupied by GDP or GTP. Normally it is occupied by GDP. When the hormone binds to its receptor, GDP is displaced by GTP. Okay, this is active stage. Okay, the GTP bearing alpha subunit dissociates from the beta and gamma subunits. The alpha subunit goes and binds to adenylate cyclase. Adenylate cyclase is activated. Active adenylate cyclase produces CAMP. Here you could see cell membrane is there, receptor is there. GTP protein in the inactivated GDP stage is there. Then adenylate cyclase is also freely situated there. Okay. Then hormone, some sort of hormone is attached to the receptor. Then there occur some reactions which change the GDP stage to GTP stage. So that will activate this G protein, and one segment, this alpha subunit, will travel into the adenylate cyclase effector. So alpha subunit has intrinsic GTPase activity which slowly hydrolyzes GTP into GDP. So alpha subunit has intrinsic GTPase activity which slowly hydrolyzes GTP to GDP. When GTP is converted to GDP, the alpha subunit dissociates from the effector. Effector in the sense adenylate cyclase. Okay, it goes back to and reassociates with alpha and beta subunits as per this picture. Okay. Then the G protein which stimulate the effector are known as stimulatory G proteins or GS proteins. There are inhibitory G proteins or J proteins also which inhibit the effector. Then next, CGMP. So the hormones using CGMP as a CGMP. Next, CGMP. The hormones using CGMP as a second messenger include atrial natriuretic peptide, nitric oxide. CGMP is formed from GTP by guanylate cyclase. Okay, GTP is there, PPI is there, guanylate cyclase, CGMP phosphodiesterase, then GMP is produced. Two forms of guanylate cyclase are known: membrane-bound form and cytosolic soluble form. ANP activates the membrane-bound form. NO activates the cytosolic form. AMP activates the membrane-bound form. ANP activates the membrane-bound form. NO activates the cytosolic form. Granulate cyclase activity is present in the cytoplasmic portion of ANP receptor. This is switched on when ANP binds to its receptor on the cell surface. CGMP is second messenger for NO also. NO is the only hormone known so far which is a gas. NO is synthesized from arginine by nitric oxide synthetase or NOS. Guanylate cyclase activated by ANP and NO converts GTP into CGMP. CGMP activates protein kinase C. Protein kinase C phosphorylates some target proteins. This produces the biological effects. Cell membrane is their guanylate cyclase domain. Atrial natriuretic peptide, GTP, GGMP, CGMP, ANP receptor, protein kinase G, phosphorylation of target proteins, biological effect, soluble guanylate cyclase. Nitric oxide synthetase has three isoforms: INOS, NNOS, and ENOS. INOS is inducible and is present in macrophages and neutrophils. NNOS is present in neurons. Then ENOS is present in endothelial cells. NO synthesized in phagocytes aids in the destruction of phagocytosed cells. NO synthesized in neurons act as a neurotransmitter. NO formed in endothelial cells diffuses into adjacent smooth muscles. It causes vasodilation by relaxing the smooth muscles. Then Ca2 plus and other phosphoinositides. Hormones using Ca++ or phosphoinositides as second messengers include oxytocin, gastrin, cholecystokinin, epinephrine acting through alpha-1 adrenergic receptors. Binding of these hormones to their receptors activate phosphoinositidases. Phosphoinositidase is a membrane-bound enzyme. It is also known as phospholipase C. Signal is transmitted from the receptor to phospholipase C by a G protein. Activated phospholipase C acts on phosphatidylinositol 4,5-biphosphate PIP2. PIP2 is present in the cell membrane. It is formed by phosphorylation of phosphatidylinositol. Then phospholipase C hydrolyzes phosphatidylinositol 4,5-biphosphate PIP2 into inositol triphosphate and diacylglycerol DAG. Phospholipase C hydrolyzes phosphatidyl inositol 4,5-biphosphate PAP2 into inositol triphosphate and diacylglycerol DAG. Then phosphatidyl inositol is shown. Next, IP3 releases calcium ions from its intracellular stores endoplasmic reticulum. Calcium ions combine with calmodulin and activate calmodulin kinase. DAG activates protein kinase C. So this activated calmodulin kinase and protein kinase C cause phosphorylation of some target proteins. The phosphorylated proteins produce the biological effect. Here we could see a picture which shows all this. Hormone is there, receptor is there, some sort of activation will be happening. G protein is there associated with that. Phospholipase C is there. Then phosphatidyl inositol 4,5-biphosphate is produced. Inositol triphosphate, endoplasmic reticulum. From endoplasmic reticulum, calcium is released. Diacylglycerol and IP3 we mentioned earlier. So calmodulin kinase, protein kinase C, phosphorylation of target proteins, then biological effects are produced. Tyrosine kinase. The hormones acting via tyrosine kinase include insulin, growth hormone, prolactin. These hormones act by phosphorylating tyrosine residues of some target proteins in the cell. Most of the growth factors also act by tyrosine phosphorylation. For example, insulin-like growth factor IGF-1, insulin-like growth factor 2 IGF-2, nerve growth factor NGF, epidermal growth factor EGF, platelet-derived growth factor PDGF. So receptors for hormones acting via tyrosine kinase are transmembrane. The extracellular portion of the receptor has got the membrane binding domain. The receptors for hormones acting via tyrosine kinase are transmembrane. The extracellular portion of the receptor has got the membrane binding domain. The cytoplasmic portion has got tyrosine kinase domain. When the hormone binds to the receptor, tyrosine kinase domain become active. Tyrosine kinase phosphorylates some tyrosine residues in the receptor itself. The receptor become autophosphorylated. The autophosphorylated receptor phosphorylates the tyrosine residues of some target proteins. The target proteins become active and produce the biological effects. An example of this subgroup of hormones is insulin. Insulin receptor is made of 2 alpha chains and 2 beta chains. The alpha chains are linked to each other by disulfide bonds. The alpha chains are linked by beta chains by disulfide bonds. The alpha chains are entirely extracellular. The beta chains are partly extracellular, partly membrane embedded and partly cytosolic. So the alpha chains possesses the insulin binding site. The cytoplasmic portion of beta chain possesses tyrosine kinase domain. The alpha chains possess the insulin binding site. The cytoplasmic portion of beta chains possesses tyrosine kinase domain. The tyrosine kinase domain is normally inactive. Binding of insulin to the alpha chain switches on the tyrosine kinase activity of beta chains. Active tyrosine kinase phosphorylates some tyrosine residues in the beta chains. The receptor becomes autophosphorylated. So insulin autophosphorylated insulin receptor. The autophosphorylated receptor phosphorylates tyrosine residues of some target proteins. These include insulin receptor substrate 1 IRS1, insulin receptor substrate 2 IRS2, etc. This initiates a cascade of reactions culminating in the varied biological effects of insulin. Then regulation of hormone secretion. Minute amounts of hormone produce profound biological effects. Therefore, regulation of hormone secretion should be very precise. Hormone secretion should increase when its biological effect is required. It should decrease when the desired biological effect has been achieved. Then hypophyseal endocrine axis is one regulatory cascade. Hypophyseal endocrine axis. This is involved in regulation of several hormones. Feedback inhibition is another important mechanism. A rise in hormone concentration causes feedback inhibition of hormone secretion. As an example, secretion of thyroid hormones is regulated by both the mechanisms. When there is need for thyroid hormones, hypothalamus secretes thyrotropin releasing hormone, TRH. TRH stimulates anterior pituitary to secrete thyroid stimulating hormone TSH. TSH acts on thyroid gland and stimulates the secretion of thyroid hormones. Raised blood level of thyroid hormone cause feedback inhibition of TRH secretion, TSH secretion of thyroid hormones. So hypothalamus is there, which secretes some sort of things. Thyrotropin releasing hormone, anterior pituitary, thyroid stimulating hormone, thyroid gland, thyroxine, and triiodothyronine. In some cases, feedback regulation is exercised by some metabolite affected by the hormone. An example is regulation of parathormone secretion. A decrease in plasma calcium concentration evokes PTH secretion. PTH initiates a series of reactions leading to a raise in plasma calcium concentration. Raise in plasma calcium level causes feedback inhibition of PTH secretion. Parathyroid gland, parathormone, plasma calcium level increase. Assay of immune hormone concentrations. Hormones are present in blood in very minute concentrations. Usual techniques are not suitable for measuring hormone concentration, yet measurements are often required for diagnosis of endocrine disorders. In the past, hormone concentrations were measured by bioassay. Biological effect of the hormone was measured in live animals or in tissues. This lacked sensitivity. Modern technologies are generally based on antigen-antibody reaction. These immunochemical techniques are quick, accurate, and sensitive. The common immunochemical techniques are radioimmunoassay, enzyme immunoassay, chemiluminescence immunoassay. Radioimmunoassay (RIA). Antibodies are prepared against the hormone. The hormone is labeled with a radioactive isotope. Labeled hormone, unlabeled hormone, and the antibody are added to a test tube. The amount of antibody is kept very low. Labeled and unlabeled hormone compete with each other to bind to the antibody. The degree of binding depends upon their relative concentrations. The amount of antibody bound to labeled hormones is quantitated by measuring radioactivity. Radioactivity emitted by iodine 125 can be measured by a gamma counter. Barely, the steps are a series of tubes marked S1, S2, S3, S4 are standards and U unknown are set up. A fixed and relatively small amount of antibody AB is added to each tube. A fixed amount of labeled hormone AG is added to each tube. Unlabeled hormone AG is added to tubes marked S1, S2, S3, S4 each in increased amounts C1, C2, C3, C4. Patient serum having an unknown amount of unlabeled hormone CU is added to each tube marked U. Then antibody, we could see this antibody. Amount of antibody, amount of labeled hormone, amount of unlabeled hormone, amount of hormone in patient's serum. The tubes are incubated for a fixed period. AG and AG star compete with each other to bind to the limited amount of AB. The AB combines with AG and some with AG. The tubes are centrifuged. AG, AB and AG star AB complexes settle at the bottom. So amounts of AG, AB and AG star AB complex in the tube depends upon amounts of AG star and AG. If the amount of AG is less than that of AG star, less AG-AB complex will be formed than AG-AB complex. As the amount of AG increases, more AG-AB and less AG star AB complex will be formed. AG star AB complex is radioactive while AG-AB complex is not. The concentrations of AG-AB complex in different tubes will be different. Its concentration will be inversely proportional to the concentration of AG. Since antibody concentration is low, some unbound AG and AG star will also remain in each tube. The supernatant containing unbound AG and AG star is removed. Radioactivity is measured in each tube. A calibration curve is prepared by plotting radioactivity in S1, S2, S3 each against the concentrations of AG, C1, C2, C3, etc. The concentration of hormone in the patient's serum CU can be read off from the calibration curve. Radioactivity, hormone concentration, calibration curve, we could see. Enzyme immunoassay, EIA. The principle of EIA is similar to that of RIA. Instead of an isotope, an enzyme is used as a label. The common enzymes used as label are peroxidase and alkaline phosphatase. Instead of measuring radioactivity, the enzyme concentration is measured. For measuring enzyme concentration, substrate of the enzyme is added. The amount of product formed is determined after a fixed interval. A common form of EIA is enzyme-linked immunosorbent assay, ELISA. In ELISA, the antibody is mixed on a solid support. The solid support may be wells molded on the plastic plate. Here you could see an ELISA plate with wells molded on it. Antibody concentration in the wells is much higher than antigen hormone concentration. The wells are labeled S1, S2, S3 like that for standard solutions and labeled U for unknown patient's serum. Then steps are standard hormone solutions in increasing concentrations are added to the wells marked S1, S2, S3. Patient serum having an unknown hormone concentration is added to the well U. Hormone molecules bind to the fixed antibody molecules. The second antibody, recognizing a different epitope of the antigen hormone, is tagged with an enzyme. It is added to each well in a relatively large amount. The enzyme linked antibody also binds to the hormone. One epitope, one antibody, second epitope, enzyme, second antibody. Complex. The unbound enzyme linked antibody molecules are washed off each well now contain complexes of the fixed antibody, the antigen, and the enzyme linked antibody. The fixed amount of substrate of the enzyme is added to each cell. The enzyme converts the substrate into a colored product. Antibody fixed in well, antibody added binds to antigen added and binds to antibody, substrate plus product. Enzyme linked, substrate added converted to product. Enzyme linked antibody added binds to antigen. After a fixed incubation period, intensity of color absorbance is measured in each well. The absorbance is proportional to the enzyme concentration. The enzyme concentration is proportional to the hormone concentration. A calibration curve is plotted here too, and the hormone concentration in the patient's serum can be read off from the calibration curve. Here you could see that. Then chemiluminescence immunoassay, CLIE. CLIE is based on chemiluminescence, emission of light driven by a chemical reaction. Principle of this is similar to that of EAA. Instead of an enzyme, a chemiluminescent substance is used as a label. Chemiluminescent substances include luminol, isoluminol, actinium esters. The chemiluminescent substance is usually oxidized to form an excited intermediate. When this intermediate returns to the ground state, it releases photons. The resulting emission of light is measured. Luminol plus H2O2 peroxidase 3-aminophthalate H2O plus light. Luminol can be used as a label. The reaction will occur on addition of H2O2. Luminol and peroxidase before adding H2O2. Chemiluminescence after adding H2O2. Chemiluminescence is measured by a luminometer. This is extremely sensitive, quick and relatively economical. It is linear over a wide range of concentrations of one light. Short life of this reagents is fairly long. So that's all about this topic. Thank you for listening, and I need to mention a very important point that the slide is not mine. I have just taken this from Google. So I thank the creator of this PPT, and I'm just using this for study purpose. Thank you.